Right, okay. Right, okay. Send me the answers. Right, okay. Okay, let's discuss. So it says smoking A stimulates the secretion of mucus by the goblet cells in the respiratory tract. So I talked about some of the respiratory defenses, right? So this mucus secretion plus cilia, right? Mucus with the cilia is a defense, right? So when what component actually stops the cilia from working properly, inhibits the ciliary action? Hydrogen cyanide, if I remember, hydrogen cyanide stops the cilia, right? So when mucus is there without cilia, that's so bad, right? Why? The mucus is going to get accumulated, right? So always mucus is good only if you have a way to remove that mucus. How do you remove the mucus? By the ciliary action. Cilia නැත්තම් මේ අයින් කරන්න වැඩිපුර mucus ටික, it's going to be a problem. So smoking, yes, stimulates the secretion of mucus. That is true, right? Causes tuberculosis. Not directly, right? I mean, you can't say it causes tuberculosis. Tuberculosis is caused by Mycobacteria tuberculosis. That's a bacteria, right? So if you know too much, then it can be confusing as well. So you might know that smokers have a higher chance of having tuberculosis, right? So that's just the risk, but it does not cause tuberculosis, okay? Decrease oxygen transport in blood. How does it decrease oxygen transport in blood? By what component? By the carbon monoxide. Carbon monoxide binds to the heme group irreversibly. So the amount of hemoglobin available to transport oxygen decreases because it's irreversible, and also it just occupies the heme groups, right? So yes, AC, okay. Inhibits the action of cilia, yes. Reduces heartbeat, no. Nicotine in cigarette smoke actually increases heartbeat because it has sort of a what we call as sympathetic nervous system stimulation, right? Sympathetic nervous system stimulation sort of function is there. It's going to increase heart rate. At the same time, do what? Constrict blood vessels. That is going to increase what? Yes, it's going to increase blood pressure. Blood pressure increases. So that is going to sort of give you some additional boost, right? A bit of energy boost for a very short time. So that is sort of the addictive nature of nicotine in cigarette. Okay, okay. Right. Let's move on to molecular. Right. I didn't upload the tutorial yet because only the question part of the tut I was able to complete. But anyway, I'll send to you so that you can refer right now. But only up to DNA replication is there in terms of diagrams, okay? Only up to DNA replication. So it's not the final, but it contains all the questions, okay? It contains all the questions, but not all the diagrams that you need. To the chat, I'll send it. See if you can open it, still sending. Right, okay. So check if you can open it, then we can move on. So I'll just go through what we talked about, prokaryotic DNA packaging, right? Prokaryotic DNA packaging. So you know what are the differences between prokaryotic DNA and eukaryotic DNA? You know this, right? By this. So we consider them to be circular DNA. Prokaryotes have circular DNA, and they only have, so this circular DNA forms only one chromosome. Only one chromosome. Only one chromosome, and also it's not protein bound. Proteins associated with DNA, non-protein. Protein associated with DNA is absent, right? Non-protein bound means no histone. That's what we mean, right? There are no histone proteins in prokaryotic, what? Prokaryotic chromatin, right? There's no histone. Okay. So when we talk about the packaging, initially you have this basic circular chromosome that is given the size that is about 350 micrometers. That is completely sort of what we call as full digarro, completely straightened out. Straightened out means not coiled at all. Then the first stage is it's going to sort of so only there are two stages. So it's going to coil. Step one is it's going to coil, right? It's going to coil around this sort of a protein scaffold like this. This is the coil stage. And the next stage I won't draw, right? It's called the supercoil. So the size is decreased. Size decreases. So the coiled or the folded. So this is coiled slash folded stage. What is the size given? Uh, the size, yes, 30 micrometers, I think. 30 micrometers. So what is this in the center? What is this in the center? Yes. This is called the core. Core. Right? The core consists of what? The core consists of ribonucleic acid, RNA, and proteins. Okay? There is the composition of the core. You need to remember that. And then you need to know what are the functions of the core. Right? I'll tell you when we talk about the supercoil stage. What is going to happen in supercoiling? These are called individual loops. Right? Individual loops. After they get condensed even more, they actually get smaller. Okay? They actually get smaller. Right? Then thereafter they are called what? Domains. They are called domains. So, right, after being supercoiled, these individual loops are called what? Domains. Okay, they are called domains. So, what are the functions of this core then? Right? So it acts. So it prevents these sort of the loops from rotating. And also, as you know, your prokaryotic cells doesn't have a what? Doesn't have a nucleus. So this chromosome is just available in the cytoplasm, right? So it has to be sort of in what we call as the nucleoid area, right? In the nucleoid area, the cytoplasm, because these prokaryotic cells doesn't have a nucleus. So the core attaches this chromosome into the cluster membrane. That is one of the functions. And then it acts as a barrier for rotation, I told you. And it allows an independent nature for these domains. It gives an independent nature, right? So whenever you need to sort of get some information from this DNA molecule, for example, the bacteria, so think it's a bacteria, it's going to synthesize a new protein, right? It's going to synthesize a new toxin, right? It's a pathogenic bacteria, it's going to produce a new toxin. So when it wants to make that, it doesn't need to relax this whole DNA molecule, so the whole chromosome, in order to get that small part, right? Think that the information is here in this small part of this domain, right? In order to read that, the bacteria doesn't need to relax this whole chromosome. It can independently relax each of these domains separately. That ability is given to it by the core, right? So if they ask you, what are the functions of the core? It acts as a barrier for rotation, attaches the chromosome to the membrane, um, and allows the domains to relax and supercoil independently. Those are the three functions. Understood? And what will happen if you remove the core? It will lead to the loss of independence of the loops. Okay, clear? Yes. Okay, right. So that is prokaryotic DNA packaging, which is very simple. Then we can move on to eukaryotic packaging, which is much more complicated. Why? Why is it much more complicated? Several reasons. Eukaryotic cells have so much more DNA than prokaryotic cells. So much more DNA. And that DNA is associated with histone proteins. And there are so much genes in there. So you cannot pack all the DNA in a eukaryotic cell into one chromosome like a prokaryotic cell, one big chromosome like that. No, you have to have several chromosomes, right? You have to have several chromosomes. And even if you make several chromosomes, you have to, you have to very tightly package the DNA, otherwise there won't be enough space. Especially it will be a problem when if you didn't package the DNA properly, it will be a problem in what situation? If eukaryotic cells didn't have proper packaging. When is it going to be a problem? It's going to be a problem during cell division, right? During cell division, if the chromosomes are not small enough and properly packaged enough, you can't efficiently separate them properly, right? So you have to have very compact chromosomes that are tightly packed, especially during cell division. Okay. So come to the resource book, page number 3, architecture of eukaryotic chromosomes. So what are the special things? Eukaryotic chromosomes have a large number of histones and they make a complex known as chromatin, right? True chromatin, I forgot to mention this. True chromatin is only found in eukaryotic cells, right? We call chromatin when what? When it forms our bead on a string appearance, right? DNA wrapped around 8 histone proteins, beads on a string. So your DNA comes like this, wraps around this complex twice like this, and then continues. So this is what we call as what? Anybody know what the name of this complex is? DNA molecule wrapped around 8 histones. It's given in the resource as well. It's called nucleosome. Nucleosome. So there's nucleosomes continuously one after the other. It gives the appearance of a like a necklace, right? Beads of a necklace connected by like a string, right? Beads on a string appearance. Then you don't need to mention this heterochromatin and euchromatin right here. I had a diagram for that. Give me 1 minute. So based on the how tight your DNA is packaged, you have two sort of what we say as two types of chromatin, as heterochromatin and euchromatin. Ah yes, there's a diagram. Okay. So I'll come to that after we have discussed the packaging, normal packaging. Okay. So there are how many levels of eukaryotic DNA packaging? 4 levels. I'll start screen sharing. Okay, follow with the resource. Okay, so they say the first level is what? That is the nucleosome level. The double helix winds around a complex of 8 histone proteins and they look like beads on a string, right? Adjoining beads are connected by a stretch of DNA called the linker DNA. Yes, I forgot to mention that. The sort of the string of DNA between two nucleosomes is called linker DNA. Very, very important. Okay, linker DNA. So can you see? This is a nucleosome and this is the linker. Okay, so this is a electron micrograph showing this first level. First level. The second level, the nucleosomes twist and pack in a spiral fashion to form a chromatin fiber 30 nanometer in diameter. Remember the length. Okay. So initially, what is the length of this based on a string appearance? It's I think 10 nanometers, right? Yes, 10 nanometers. Now it sort of spirals it spirals once, right, to form the 30 nanometer thick string. Okay. Not a string. Now it's a fiber. We call it a fiber. Okay. Right. Third level, there's a protein scaffold, like a platform or like a, let's say, composed of protein. It's like a stage, right, for the proteins to sort of properly. To sort of what we say as get convoluted, right? So those are called loop domains. Loop domains. So now, because this 30 nanometer, so this is the 30 nanometer, okay? This fiber has 30 nanometer diameter. When it sort of convolutes like this, forming loop domains, its thickness becomes 300. Thickness becomes 300. Final level is the scaffold folds. Can you see? The scaffold itself forms. This is the scaffold. Red color is the scaffold. The scaffold starts to fold. Okay, the scaffold starts to fold. Therefore, the thickness will increase to 700 nanometers. So that will actually become this chromosome. So, what is this other chromosome? Or what we call as what is the other what is this? It's the other sister chromatid, right? So how did we get another sister chromatid? Right. This is why. When do we have sister chromatids? Do we have sister chromatids all the time in a cell? Sister chromatids. When do we have sister chromatids? Yes. So in cell division, you have sister chromatids. Why? Because DNA underwent doubling. What do we call as DNA copying? Making two copies? DNA replication. So DNA replication has happened. So you have two DNA sister chromatids. And also, what is the other logic? When a cell is not dividing, what stage of DNA packaging do you have? When a cell is not dividing, what stage of DNA packaging do you have? Do you have this second stage, third stage, and the fourth stage in a non-dividing cell? No. You only have only this. You only have the chromatin level. You only have the first beads on a string appearance, right? So that's why when you draw a nucleus, right, we just draw it like a ball of string, right? Because in a non-dividing cell, you only have chromatin, right? So the DNA undergoes all this packaging to prepare for what? To divide. Okay, have that in your head, right? I'll show you the sort of the my tute. I have some steps there. So this is the nucleosome beads on a string appearance. That's in your. This is a bit clearer diagram. It's in your resource. This, right? You can see the levels, right? The normal DNA. Then you have beads of string appearance. Then you have the protein scaffold. And finally, the scaffold folds to form the chromosome. Okay, clear, right? That is all about eukaryotic DNA packaging. I'll come back to what is this. What we missed. What is heterochromatin and euchromatin, right? So this is a feature that you can see in the chromatin, not in chromosomes. So when tightly packed, you can't see these heterochromatin areas and euchromatin areas. Okay, only when you have the first level, that is the beads on a string appearance, that is in a non-dividing cell, you have this euchromatin and heterochromatin, right? So euchromatin is lightly packed DNA. Can you see? This is a heterochromatin. This is hetero. This is euchromatin. Heterochromatin, can you see? Densely arranged. Euchromatin, loosely arranged. It's like when in a book rack, if you have, sorry, in a book rack, if you arrange the books very too tightly that you can't even take them out, then those books don't get used that much because the people can't access them. But if the books are sort of lying around everywhere loosely, people tend to read them very often, right? They are commonly used. So, on the other hand, the most commonly used books are always lying here and there, right? But the books that you don't use at all are inside cupboards or tightly packed. It's like that. Heterochromatin are the DNA contains sort of the genes that are not very commonly required. Yeah, those genes are not commonly required. But the euchromatin contains genes that are very commonly referred. That you read those genes very commonly. So they cannot be tightly packed. So they have to be sort of loosely lying there. So the mRNA and other things can come and sort of read them easily. Understood? Any questions? Yes. Okay. Any questions? I should put this in the tube test. Okay, right. Do we have any MCQ from? Yes, I think we did this one. Yeah, nothing from packaging. I was not able to find anything from packaging. Right. We can move on to DNA replication. Okay. Any questions up to there? Up to that point? Yeah. Okay. We can start. What is the definition of DNA replication? A process which copies a double-stranded DNA molecule to produce two identical copies. Right? You always have to have two-stranded DNA in order to do DNA replication because what we call as a semi-conservative process, right? It's what we call as a semi-conservative process, right? Because one, so initially you have double-stranded DNA, no? They will be separated, like torn apart. Then using each as sort of like the blueprint, right? Using it as the original, another DNA will be sort of synthesized opposite it, right? So the new DNA double strand you get has what? Has one new strand and one old strand, right? So it's not an entirely brand new DNA that you are making, right? It's not an entirely brand new DNA that you're making, right? In the new DNA double strand, one strand is the old one, only one is brand new. Got it? All right. So replication, I think it's here about this semi-conservative process. Yes, ignore the others. This is what happens in your normal DNA replication. So you have the old strand, and using the two old strands as the template. That's the word I forgot. As the template, you will make two strands. Okay. Understood? Right? Okay. Then what do they say? DNA replication process is basically similar in prokaryotes and eukaryotes, but there are differences in the enzymes, right? At the end, we'll compare what are the differences between prokaryotic and eukaryotic DNA replication. And why is it? Because eukaryotes have chromosomes and we have histones, and prokaryotes don't have histones, and they are usually circular molecules without very tight package. Okay. So there's some things about how the important it is. So these are things you know, right? You can read by your own. Nothing important here, right? Except this one maybe. Um, DNA replication is a very accurate process, right? Accuracy is very high. We'll see how this accuracy is maintained. It's called the proof reading mechanism, right? Yeah, very accurate. The enzyme that is making this is very accurate, right? So if there is some mistake, it can cause a change in the DNA that is called a mutation, right? So this is, I don't need to explain this, but please go through it and highlight. Okay, so in your syllabus, you are discussing the replication process in prokaryotes. Then at the end, we are just going to discuss what are the differences. That is basically how it's going to happen. Okay. Um, Step one in order to do anything, what do we have to do? We have to get access, right? We have to get access. That accessing has to be done by one person, right? So this is a sort of like a factory, right? DNA replication requires a team of enzymes. It's a team of enzymes. One person cannot do this. It requires a team of enzymes, right? And that team has different people that are specialized to do different things, right? Step one is you have to separate the double helix, right? So that is done by, sorry. So as you know, DNA synthesized on old DNA strands. So they are used as templates. So to use DNA as a template, you have to break the double helix. You have to remove the two strands away from each other by breaking what bonds? Breaking what bonds? Yes, by breaking what bonds? What are the bonds that are connecting the two DNA strands together? What are the bonds found between the? What are the bonds found between what? The two strands. What are they? Between the two nitrogenous bases. Basics. What are the bonds found between the two nitrogenous bases? Yes. Come on, anyone? Base pair rule, ATGC. No. Adenine connected to thymine by two what bonds? Guanine connected to cytosine by what bonds? Hydrogen bonds. Okay. Okay. Right. You have to break the hydrogen bonds. So before that, we have to decide where we are going to start DNA replication from. Right? We have to decide where we are going to start. So that place is called origin of replication. Origin of replication, it is here. Right? The decopying process starts at the origin of replication, or you can call it as ORI, or ORI site. We call it ORI site. Okay. What is the definition of ORI site? It's a specific sequence of DNA to which proteins that initiate DNA replication bind. Okay. Origin of replication, ORI. So I'll show you ORI. So we are talking about this now. We are talking about a prokaryotic cell. So this is your ORI, right? At ORI, you will start DNA replication. Got it? So we have sort of, this is like a electron microscope image transmission. So this is, I think, is this, these two are the original circular chromosome, and this is the new strand that is being synthesized. Okay. That is the ORI. So remember, replication begins at the origin of replication called ORI. Okay. Then, since the enzyme synthesizing our new DNA can move only in one direction, 5' to 3' direction. Why is it only in 5' to 3'? These are basics. Go back to nucleic acids and C. Because why? Phosphodiester bonds are formed from the phosphate group that is bound to the 5th carbon, and between what? With the OH bound to the 3rd carbon. So what is the direction? Downwards, right? 5' to 3' direction. So the molecule, the enzyme that is adding these new nucleotides to create the new strand also moves in the normal direction, that is 5' to 3'. That is another information you have to remember. And what's the other problem? So yes, when you take a DNA strand, sorry, when you take a DNA strand, what is the issue? You have two strands, no? You have two DNA strands. So you have one that is running in 5 prime to 3 prime, yes. But his complementary strand runs in 3 prime to 5 prime. 3 prime to 5 prime. So what is going to happen when we open up, right? When we separate the two strands at the origin of replication, this guy, right? Why? So this is the 5 prime end of the template. Parent, this is the 5 prime end of the parent, and this is the 3 prime. So what does that mean? This is the 5 prime end of the new strand called the, so it's the new strand. So in this direction from 5 prime to 3 prime, when the sort of the enzyme that is making this new strand, it can move easily from this 5 prime to 3 prime direction. But what's the problem? The DNA new synthesis has to happen in this direction as well, right? So you're moving in this side, no, you are sort of separating the DNA, parental strands, and you are sort of going forward. But what's the issue? In this strand, in the below strand, this is 3 prime and this is 5 prime. So if you want to make a DNA strand in this direction, what is going to be a problem? This is 3 prime and this is 5 prime. So can you make a DNA molecule from 3 prime to 5 prime? No. So what is it going to do? It has to be like a typewriter. It has to sort of make some DNA and go to the front. Make some DNA and go to the front. Like that. It has to jump like this. Can you see? It's going to make a small DNA strand from 5 prime to 3 prime direction. And sorry, take this one. It's going to make a small DNA strand from 5 prime to 3 prime direction. Then it's going to jump forward to here. Then it's going to make another fragment here. Then it's going to jump to here. Right? It's like a typewriter. It's going to make a small strand, then jump. Make a small strand, then jump. Get it? That is called the lagging strand. Got it? Anyone? What's the issue? Okay, you have got the answer. Okay, right. What's the issue? The issue is we have to make two DNA strands, right? We have to make two DNA strands. So one DNA strand has no problem because it is going to be made in the proper direction of 5 prime to 3 prime. But the other one is going to have the problem because the DNA making direction is going to be 3 prime to 5 prime. That is going to be a problem. So the the enzyme that is making the new strand has no option but to come here and go in 5 prime to 3 prime direction. So if it's going in this direction, it's okay. But what is the issue? We have to go in this general direction. Right? This is the direction that the DNA making should happen. But if you are traveling this way, it's a problem. So what is it going to do? It's going to make a small strand and then jump. It's going to make a small strand and then jump. So in the end, it's moving, but bit by bit. Right? Okay, let's continue. So one strand is synthesized continuously. The continuously synthesized strand has a name called the leading strand. And the one that is not continuously synthesized but made in small parts is called the lagging strand. And those small parts of the lagging strand are called what? Okazaki fragments. Okazaki fragments. Okay. Okazaki fragment. So this is not a very good diagram, right? So you have here in a prokaryotic cell how the origin happens and how when the so this is the origin. So green is the origin, and this orange is the end. So when it has traveled from green to orange, what does that mean? It has gone a full cycle, and a completed new copy is completed. Right? That is the process. Any questions? So let's read the resource and come back to more diagrams, right? So number of enzymes and other proteins are required, and all these people, it's like a workforce, like a team. So where should they start work? At the origin of replication. They all assemble at the origin of replication. What are there's a list. We have helicase, topoisomerase, primase, DNA polymerases, right? It's a family of enzymes, and DNA ligase. Okay. Then there are enzymes. So then there are proteins. So all these were enzymes, and there are proteins such as single strand binding proteins. Okay. So what you say as the first man to get involved in the process is called helicase. What is the function of helicase? They unwind. So it's a twisted ladder. DNA is a twisted ladder. It unwinds it so now it's a bit more free. Then what does it do? It separates the DNA strands using ATP energy. So it requires ATP. So it involves the breaking of the hydrogen bonds. This is important for exposing the two strands to be used as a template, right? So can you see DNA helicase is the first person that is coming and separating the two strands. Okay, helicase. Then topoisomerase. Topoisomerase is sort of after DNA helicase starts his work, topoisomerase runs ahead, and he's going to solve a problem that is happening in front. He's going to solve a problem that is happening in front. What is the problem? Imagine you have a rope that is formed of two small ropes, twisted rope, right? Twisted rope. So imagine you are separating the two small ropes by pulling on the two ends, right? When you're pulling on the two ends, what is going to happen? It's going to get tight at one side, right? It's going to get tight at one side because you're sort of pulling apart. Topoisomerase is going to run ahead, and what is it going to do? It's going to relax. So I think there's a diagram of what the topoisomerase does. Topoisomerase. It's not given here, the topoisomerase. So if this so this is the place where the copying is happening. So this is the place where copying is happening. Topoisomerase is sort of far ahead, around here, right? He's relaxing the two strands. He's relaxing. How is it relaxing? Why is it not given anywhere? Topoisomerase. I think this is topoisomerase in this diagram, right? Can you see? Ahead of the strand, relaxing the strands, topoisomerase. Okay, so how does it work? It says untwisting the strands, so that causes further twisting and strain on other places. What does it do? It introduces breaks on one or both strands, twists the molecule to relieve the strain, and reseals. What is it going to do? It turns it in the opposite direction. To relieve the tightness, and then again seals it. Got it? So topoisomerase. Then we have the protein that is called the single strand binding protein. That is there. So imagine these two strands are like magnets, two magnets, right? Once you have separated them, they like to get closer and then form hydrogen bonds, right? So if you don't sort of hold them back, they will slowly get together and bind again. So single strand binding protein prevents them from becoming double again. How does it do that? It just binds and stays there, right? Okay, bind la. Can you see? This is a single strand binding protein. This is a single strand binding protein. They just bind to the separated strands and sorry, prevent them from joining back together. Simple. Okay. They also have a stabilizing function. Okay. They also have a stabilizing function. Stabilize. Okay. So why is this important? If the two strands connect with each other again, can you use them as templates? No. You can't use them as templates. Okay. All right. Then we come to the actual DNA copying part. This is Apita, we were just starting the process. We were just separating and stabilizing and preventing reconnection. We were doing all those. So now we come to the proper copying. But it's not simple as that. Copying is not simple as that. Because let's say, so the main guy, main sort of the worker that is actually adding new nucleotides is called DNA polymerase 3. Okay. So this is him, right? Uh, this is the guy, right? DNA polymerase 3 is the main man, right? The main role. But think of him as a bit shy, right? So he doesn't like to go and start work, right? He doesn't like to go and start work, right? He needs a small friend to go and start the work for him. Then he will come and he will continue the work. Got it? That's how this DNA polymerase 3 is, right? He doesn't like to go and start the work, but once someone has started the work a little bit, he's going to go and he's going to take over and he's going to make it, right? That's how DNA polymerase 3 behaves. So who is the person that is going to go and start the work? That is another polymerase, but not a DNA polymerase. Okay. It's a polymerase, but not a DNA polymerase. Right? Who is it? It's not given here. Who goes and starts the process? Right? It's a RNA polymerase. Okay? See, it's an RNA polymerase. Can you see? So that RNA polymerase has a special name called the primase. Primase. Right? So they say DNA polymerase can add nucleotides only to 3' end of an already formed nucleic acid chain. So he doesn't like to, DNA polymerase doesn't like to go and start from the start from the beginning. He has to have a very small chain of DNA, sorry, very small chain of nucleic acids already there for start work. So who adds that small chain of nucleic acids? Primase. Primase is an enzyme. It's actually a RNA polymerase. Very, very important. It's not a DNA polymerase. Primase is a RNA polymerase. Therefore, this small fragment of nucleotides or nucleic acid added is what? Is it a DNA piece? Yes. Is it a piece of DNA? No. It's a piece of RNA because it was added by an RNA polymerase that is called RNA polymerase, also called as the primase. So primase has a short RNA primer on the DNA template forming a DNA RNA hybrid. This facilitates the DNA polymerase, specifically DNA polymerase 3. Okay, this is called initiation of replication. So now we have started the process. Now the big man can come. What is that? That is DNA polymerase can come and start the work, right? So DNA polymerase will add complementary deoxyribonucleotide to the 3' end of the primer and continue polymerization in 5' to 3' direction. That is what's happening here. So can you see in this diagram? This is the small RNA primer that was added by primase, which is a RNA polymerase. And now DNA polymerase is here, and it's going to make the strand. See, it's going from 5' to 3' direction. No issue at all. Following so far. Then, what about the lagging strand? I'll just mention, right? I told you it jumps, right? It goes in the 5', 3' direction and it jumps. 5', 3', jumps. Small pieces are made. So this is the primase and this is the DNA polymerase. So it makes this small piece 5' to 3'. But what's the issue? DNA synthesis happens in this direction, but the polymerase is traveling in this direction. So the polymerase will make this piece and he will jump. So this is called Okazaki fragment. And later, the Okazaki fragments will be joined together. Okay. And I forgot to mention something here. No, I'll mention this later. Okay. So DNA polymerase we talked about. Then let's talk a little bit about this DNA polymerase. He is 100% error free. Almost 100% error free. However, one error in, what is this number? 1 in 100,000. Lakshakata ekak errors can be there. But he has a proofreading mechanism that can correct his own mechanisms, reducing the error rate to 1 in 10 to the power 10. What is this value? 10 to the power 10 means 1 in million is 10 to the power 6, no? Billion is 9. 1 in 10 billion. 1 in 10 billion nucleotides error. I have no idea. What is 10 to the power 10? 10 to the power 10. I think 10 billion, no, whatever. Okay, that is how accurate he is. So this is the correcting mechanism or the proofreading mechanism. He identifies the mistake by himself, removes the incorrect one. By what? It breaks this bond here. It breaks the phosphodiester bond relevant, and then the proper one will be added. Can you see? G, for the G, he has added what? Guanine, he has added an adenine. So that's wrong. He will see that, he will remove it. By what? Exonuclease activity. All right. Then we come back to the primase. So we thought we forgot the primase, no? But the primase is a problem. Can you see? Why is sorry, not the primase, sorry. The primer. The primer is a problem. Why is the primer a problem? Why can't we keep the primer? Why can't we forget about the primer? Why can't we forget about the primer? Because he's... Why can't we forget about the primer? Because he's a RNA. We are making DNA, no? Primer is a RNA. So we cannot leave RNA in a DNA strand. So there comes a different DNA polymerase. You don't need the name. It's actually called POX1. It will remove the primer and will add deoxyribonucleotides instead of ribonucleotides, replacing the primer and making the fragment whole. Now, right. So we have made a DNA strand. What do we have to do? Finally, the glue comes in. The welder, right? He comes and welds, right? It's like the welder, right? He comes and welds the different pieces of DNA that was formed together by making phosphodiester bonds. That guy is called DNA ligase. Very simple. DNA ligase. Okay. So this is that whole process. This is making the leading strand. This is the first lagging strand completed. DNA ligase has come and connected the first lagging strand and second lagging strand. So DNA polymerase here is removing, in the process of removing the primer. Here, the primer is still here, but the third Okazaki is still there. Fourth Okazaki is being made by DNA polymerase. Single strand binding proteins are stabilized. DNA helicase is separating. Somewhere ahead, topoisomerase is there, relaxing the strands. Got it? This is the story. All right. Proofreading mechanism, I put this diagram in your tutor as well. By DNA polymerase, how it proofreads, right? It can identify the mistake and remove it by exonuclease activity and add the proper fragment and then continue copying. Right? This is the summary. This is the summary. Now we come to similarities and differences between prokaryotic and eukaryotic DNA. Similarities and differences. So what sort of replication did we talk about? We talked about prokaryotic replication. Prokaryotic, right? Prokaryotic replication. Now we have to relate this prokaryotic replication to eukaryotic genes or eukaryotic chromosomes and see how it's different. The basic concept is the same, right? As we mentioned, the enzymes are different. Why the enzymes are different? Because eukaryotic chromosomes are more complicated. Simply. What are the similarities? I'll highlight the similarities in blue. They use helicase to unwind, sorry, polymerase to polymerize, originated at ORI sites. Packed DNA is relaxed by topoisomerase. They move in the same manner, right? They have lagging and leading strands. RNA fibers are there. The gaps are sealed by ligase. All those are similarities between prokaryotic and eukaryotic DNA synthesis. Just read the similarities by yourself. That's good, buddy. What is that? Good. Are you taking it? Right, okay. So those are the similarities. What are the differences? What are the differences? I'll highlight them in the pink color. So the size of DNA is much larger than, I mean, size of even a single chromosome in the eukaryotes is larger than the one. I mean, they only have one chromosome. No? That is even bigger than the one chromosome that they have, one big one. So why only one oocyte, only one origin of replication is not enough. It's going to take forever if they start with only one origin of replication. So the first difference is eukaryotic cells have several ori. Prokaryotes have one ori. It's like this. Uh, can you see? In a eukaryotic cell, you have several oocytes. So you can see in this DNA, new strand synthesis is happening in several places. At the end, all these small pieces starting at different oocytes will be connected. So it's a fast process. So instead of having one team, you have several teams working at different places. So when the teams get, when the teams get close to each other, all their work will be connected together, and you will have a finished product. You will have a finished product. It's a teamwork. Okay, that's the first difference. Then the polymerases are a bit structurally different. DNA polymerases are structurally bit different. Because why? Because of those differences we talked about. We have helicase, sorry, we have DNA, sorry, we have histones in eukaryotic chromosomes, and there are other differences as well. And DNA replication in prokaryotes occur continuously, where in eukaryote it happens only in S phase. Yes. Why is DNA replication happening continuously in prokaryotes? Because they don't undergo a proper cell division like us. They don't undergo a proper cell division like eukaryotic cells. They can divide suddenly. By what division? By what cell division? It's not a cell division actually. Itapugamum, they can just divide by binary fission. Right? Prokaryotic cells just break into two pieces. So whenever they just undergo binary fission, they have to have DNA ready. So that's why continuously prokaryotes are making DNA, making DNA, making DNA. But in eukaryotic cells, we have to make chromosomes. We have to copy DNA, we have to make chromosomes, and then we have to sort of pull them apart properly. Either it is meiosis or mitosis, right? Therefore, DNA replication occurs only when? When do we have DNA replication? In the S phase. In the S phase of cell division only, you have DNA replication. Okay. Right. I think you listened enough. Let's go do some questions. This one, question number 32. Right, okay. Okay, have you sent the answers? So let's see. Uh, a function of topoisomerase is sealing the gaps of DNA strands. No, that is done by, write a review. Okay, who seals the gaps? Who is the welder? Ligase. Unwinds the double helix. Who is separator? Who is separating the two strands? Helicase. Stabilizing. Who is stabilizing? Is it an enzyme? SSB, single strand binding protein. Relaxing the strain. Yes. Who is the relaxer? That is the topoisomerase. Breaking hydrogen bonds between DNA strands. That is again helicase. Helicase does two functions. It unwinds and separates. Right? Helicase does the two functions. All right. Got it. Hmm. Hmm. All right. Right. Do this one. Just a small structure. This part. The same diagram in your resource. Okay. Right. So it's a basic question. Remember enzymes A and B. You know, state the main function of each of them. What is enzyme A? So let's just label all these. So who is B? I mean, I think it's easily identifiable. Right at the separating site. Right at the separating site, who's there? Where the two strands are getting separated. So B is definitely, yes, helicase. Then if it's helicase, who is the only one that is ahead? Yeah, ඉස්සරහින් වැඩ කරන කවුද? Who's the only one that is working ahead? Sorry, topoisomerase. This is topoisomerase. Then these are what? SSBs, no? They are just they have just holding on to the strands without letting them combine. That is SSB. So A has to be DNA polymerase. Okay, so you know the functions. Okay. Yes. This one. State the significance of the RNA polymerase in DNA synthesis. What is the name of this RNA polymerase and mention why is it important? What is the RNA polymerase? It's the primase. Primase is necessary to add the primer because DNA polymerase can only add nucleotides to the 5' end, sorry, the 3' end of an already existing nucleotide chain, right? It doesn't have to be DNA or RNA, anything, but here RNA polymerase adds a primer, which is a piece of RNA. Got it? Right. So that is all the questions that is from, they are from DNA replication. There hasn't been a, I mean, there hasn't been an essay question. So anyway, you only get half of an essay. You only get half of an essay from molecular. Always either part A or part B. So never a full essay. So DNA replication is a bit too long to be an essay, but you never know. Okay. So let's just talk about how DNA repair is done, and then let's end for the day. Okay. So DNA repair and its significance. We'll talk about what happens when DNA is damaged, right? So there are several ways that DNA can get damaged, right? So this is to be, I mean, understand clearly, this is not errors in synthesizing, okay? So these are not errors done by uh, what DNA polymerase, right? These are damages that occur after the completion of the synthesis, okay? So there can be damage by chemical agents or physical agents, right? So, and also very rarely, as you know, 1 into 10 to the power 10 times is the likelihood errors that are unnoticed by proofreading can also be there, very, very small. So these errors are all mutations, right? But I mean, you'll learn when we're discussing mutations at the end, you'll see there are beneficial, neutral, and I think harmful, or I can't remember the name. There are beneficial ones and then neutral mutations and harmful ones, right? So almost all mutations are either neutral or harmful. Very, very rarely you have a beneficial mutation. Okay. Very commonly, a mutation can lead to an abnormal cell that has abnormal behavior leading to a cancer. Okay. So they are often lethal or least produce unfavorable phenotypes. Right? So cancer can only be passed from one generation to the next, or these mutations can be passed from one generation to the next only if they occur in where? In the gametes, either in the gamete, the cells, or even in the gametes, if they are produced, then only they can be passed to next generation. So example, if you have a skin cancer, right, happening in your skin epithelium, it won't be passed to your next, it won't be passed to the next generation because it happens in your just your somatic cells, body cells, right? It's like that. So only one example is given for you, that is this UV radiation induced thiamine sort of, I can't remember the exact name. So what happens is when you have two adjacent thiamines, sorry, thymine, okay, thymine. when you have two adjacent thymine, what is going to happen? Wait, I will show you. It's actually only one of the ways DNA repair can happen, right? Uh, DNA repair. So common causes, there's chemicals, right? Chemicals can be oxygen radicals, right? Then physical can be radiation, right? UV light, things like that. So we have this one. Only in your syllabus you have this one. What is it? Two adjacent thymines. Yes. No. Yes. This is it. Yes, it's called UV-induced thiamine dimer repair. Okay, UV-induced thiamine dimer is the only mechanism that is given in your resource. What happens is when DNA is exposed to UV radiation, and if there is two thymine bases close to each other, what is going to happen? They are going to sort of get removed. Sorry, they sort of get disconnected from their cytosines, and they just connect with each other. What is it going to happen? The nucleus is going to come and going to cut away a part and replace it with the proper, like a healthy piece, right? So these are proper, right? The thymines are okay, they are not mismatched. But what happened is UV radiation has come and connected them abnormally. So the the polymerase will come and put a proper piece there that is not damaged, right? Okay. Okay, so I will add it to the tip, right? For example, UV radiation can covalently link, very important, covalently link two adjacent thymine bases, distorting, right? They are usually recognized and repaired by DNA repair mechanism before it becomes permanent. Large number of DNA repairing repairing enzymes are found. And what are the things that are done to repair, right? They do excision by a nuclease, and a polymerase will come and put the correct strand, and a ligase will come and seal it. Three people are mainly involved. What? A nuclease to cut, a polymerase to fill the gap with the proper strand, and DNA ligase to connect everything after the repair process has been done. Okay. Right? So I think we can continue with the genes and finish protein synthesis next week with the genetic code and everything next week. Okay, with the translation and transcription. And the fate of proteins. Then we can come to mutations, right? That's going to be another day, the mutations one day with the diseases. Right? Then gene technologies, that is just, that is going to take another day, even more than a day maybe, right? So yeah, I think we better talk about the genes today, no? Otherwise we might not have the time, right? Let's just talk about the prokaryotic and eukaryotic genes today as well. Otherwise we might not have time. Okay, let's talk about genes as well. So you have to be able to compare prokaryotic and eukaryotic genes, right? So you know the concept of the gene was put forward by who? I mean, he didn't know that there were genes, right? He didn't even give the word name genes, right? He gave the term hereditary factors, right? Hereditary factors determine a particular character, right? A gene determines a particular character. That's all he knew, right? So to him, they were imaginary units, and he didn't know where are they and what were they made of and how they work. He didn't know. What he only discovered was they had a effect, right? They produced the effect. So the gene definition is it's the fundamental or basic physical and functional unit of heredity. It's a segment of DNA on a specific site on a chromosome, and it specifies the sequence of RNA. What is the RNA that is made from looking at DNA? What is the RNA that is made by looking at DNA? mRNA, mRNA, messenger RNA. So let's go to DNA genes, gene expression. Right, so this is the basic story. I hope you remember DNA. Using DNA, we make an mRNA. By looking at the mRNA, a ribosome will make a protein. This is the story, right? So in prokaryotes, it's simple, but in eukaryotes, it's a bit complicated because the DNA inside the nucleus. Okay, right. Right, so we'll come there. Structure of a gene, I have to explain. There was a good diagram. Oh, yes. Okay. Right. So you can read this paragraph, right? Because we are diploid organisms, right? Because we are diploid organisms, we have one pair of genes that is coming from, sorry, one copy of genes that is coming from the father and one copy from the mother, right? Those are sort of, that's why we are called diploid, no? So we have two alternate versions of genes, right? One version from the father, one version from the mother. So those are called what? What are the two versions of a gene called? What are the two versions of a gene called? Genetics knowledge. What are the two versions of a gene called? Like different versions. So yeah, the versions is the word, right? They code for the same character, but they are slightly different. They code for the same thing, but they are slightly different because one is coming from the father and one is from the mother. They are called what? Alleles, right? So that's what they say. Pair of chromosomes coming from the parents contain the same genes. They are homologous chromosomes. You know homologous chromosomes, they are equal height ones, right? So usually prokaryotes have only one chromosome. They don't have sexual reproduction, so there's no father or mother for a prokaryotic cell. So they only have one chromosome. So they are anyway haploid, right? Then in the definition it says a gene is located on a specific site on a chromosome. What is the site of a gene in a chromosome called? The address of a gene. Where does the gene live in a chromosome? So if the chromosome is the country, a gene living in that country has an address. Which arm of the chromosome? And where in that arm does the gene live? That is called the locus. Think of it as the address of a gene. Okay. If you want to find a gene, you have to look at the locus of that gene. Right? So Alternative versions of genes that are located at the same locus on different chromosomes are called alleles. Usually, what are these different chromosomes? These are the homologous chromosomes. These are the homologous chromosomes. Because why? One pair of the homolog chromosome one part of the homolog pair came from the father, one part came from the mother. Right? In prokaryotic genes are housed in loci in the same same on the circular DNA molecule as discrete segments. Okay. Right. Okay. So then let's talk about how different genes are. Differences between eukaryotic and prokaryotic genes. At the end, I want you to compare. Prokaryotic genes, eukaryotic genes. Prokaryotic genes and eukaryotic genes. You have to be able to compare. What are the differences? So, first thing, if you take a eukaryotic chromosome, the genes are scattered everywhere. Genes are scattered throughout the chromosome. Genes are scattered throughout the chromosome. But if you take a prokaryotic chromosome, the genes are found one after the other. The genes are found one after the other. Okay, one after the other. So it's like this. If you take the chromosome like this, gene G1, I'll call G1 as the gene 1, gene 2, gene 3, like that. The genes are found one after the other, right, in a prokaryotic chromosome. But in a eukaryotic chromosome, it's much more complicated. This is the DNA or the chromosome, right? There are so many, there's a gap here, and there's the G1, first G. There's a gap here, and there's the G2. There's another gap here, there's the G3. Okay. There are what we call as non-coding DNA pieces in between genes. So the genes are not found one after the other properly. There is a difference. Okay, clear? Then, so what is going to happen in prokaryotic genes is when you are making an mRNA, so by looking at DNA, so this is DNA, this is DNA, you're going to make an mRNA looking at this. So this is called transcription. Making RNA by looking at DNA. All right. So when you are making this mRNA, it's very easy, right? So this is gene 1. This is gene 2. This is gene 3. You can make a mRNA very easily. Right? So that's what they say. They are arranged in a prokaryote. They are arranged in the same region of the chromosome, one after the other. Therefore, they can be transcribed into one mRNA. No problem at all. Simple. Right? So, what is the next step? After transcription, translation. Translation happens in the ribosomes. It is also very easy. So let's say using the first gene, you are making a protein 1, protein 1, protein 2, protein 3. Sorry, protein 3. Understand? In prokaryotic genes, it's all very simple. You have genes one after the other. You can make an mRNA very easily. Using that mRNA, you can make proteins very easily as well. Okay, that is about prokaryotic genes. But let's jump to eukaryotic genes. In eukaryotic genes, there are regions in DNA that we don't know what the function is. Okay, we don't know what the function is. It's not actually this simple. I'll draw this a bit more. It's actually a bit more complicated. Right? So like this. This is gene 1. Okay. Gene 1. But in the middle of gene 1, you have this is again gene 1. But then there's a gap here, and then there's gene 2. I'll explain this. Okay, I'll explain. So in the middle of gene 1, you have this sort of non-coding piece of DNA. And in between genes also, you have this non-coding DNA pieces. And then you have another empty part, and you have gene 3. Separating them, you have another non-coding. Non-coding means they are not coding for any proteins. Okay, I'll explain this. Eukaryotic genes are very complicated. So, eukaryotic DNA is actually very sort of mysterious, right? There are large parts of the eukaryotic DNA that we don't know what the function is. We don't know yet. Maybe they have discovered, but not in your syllabus, so I don't know, right? So maybe they have discovered, but we know that, there are parts of DNA that we don't know the functions of them yet. They are of two types, right? If they are found between genes, what are they called? They are found between genes, what are they called? Between genes, no? Intergenic DNA. Intergenic DNA. Intergenic DNA. Right? In between genes, you have intergenic DNA. But also within a gene. So this is gene 1. So this is still gene 1. In the middle of a gene, you can have non-coding parts as well. Those are called introns. I hope you remember, introns in genes is a feature of eukaryotic cells. Go to your second lesson, cellular basis. Right? Sorry, I'll stop screen sharing because it's difficult for you to see the board, no? I forgot. Really sorry. Okay. It's difficult for you to see the board, no? Yeah. So I'll explain what I have drawn again, right? So this is a prokaryotic gene. There you have genes one after the other, right? So they will be transcribed into an mRNA very easily, G1, G2, G3. And using those, you will make proteins. Protein 1, protein 2, protein 3, no problem. This is a eukaryotic eukaryotic DNA molecule. And there the things are complicated. Genes are not one after the other. There are gaps between genes. And those DNA that is in between the gaps are called intergenic DNA. And also, in the middle of a gene, you have non-coding DNA sequences. Those are called introns. Those are called introns. Okay? Introns. Then how is it going to happen? How is the transcription going to happen? How are you going to make an mRNA? mRNA will undergo a middle step, right? mRNA will undergo a middle step. So we are going to do transcription, okay? We are going to do transcription. But what is the issue? We are going to do transcription. Let's do transcription of this gene, gene 1. Let's do transcription of this gene, gene 1. When you are transcribing, right, in the middle of the mRNA, you are going to have a non-coding part. Non-coding part. That is what? That is this intron. I hope you can see. I will draw it a bit thicker. Right? In the middle of the normal mRNA, you're going to have a non-coding mRNA that is coming from where? From the intron. So this is not a proper mRNA. This is not a proper mRNA. We call this as pre-mRNA. Pre-mRNA. This is called pre-mRNA. And it has to undergo another step where we call processing, right, to become proper RNA. It has to undergo processing to become proper RNA. Then this red part will be removed and you will get a proper mRNA. Proper mRNA. Got it? If you have forgotten, what are introns and exons? What are introns? Introns are non-coding parts of DNA in genes. Write down if you have forgotten. Non-coding pieces of DNA in genes. Introns. Non-coding pieces. So, if this is an exon, sorry, if this is an intron, what are these? These also have a name. The coding pieces. They have a name. They are exons. Exons are the coding parts of a gene. Coding pieces of DNA of a gene. Anybody have any questions? Anyone? Any doubts? Nisila is here, right? Who else? Do I need to explain this again? Because I think I was screen sharing and the board was small, no? Yeah? Any doubts? Do I need to explain again? Avikshana, any questions? This is how the two types of genes work, right? What are the main things that you need to know? Main points. Prokaryotic DNA, genes are arranged one after the other. Eukaryotic DNA, genes are scattered in the DNA molecule, right? And there are non-coding DNA in between genes called intergenic DNA. We don't know the functions of them, right? It's a mystery. Maybe they have some regulatory effects. We don't know yet. They are called intergenic DNA. In the middle of a gene also, you can have coding and non-coding DNA pieces as well. Non-coding DNA pieces in the middle of a gene are called introns. Coding DNA segments are called exons. When you are transcribing or making mRNA, the introns also get converted to mRNA, but they are not necessary. So we have to process this pre-mRNA molecule to make the proper mRNA. So you can even uh, draw an arrow here. This is the intron. These are the exons, right? So this is the pre-mRNA. This molecule is the pre-mRNA. It has to undergo processing in order to have the proper mature uh, mRNA. Got it? Right? So genes, with that knowledge, we can next week move on to protein synthesis from transcription, process of transcription. You have an entire different set of proteins for that, and translation, right? I hope it's not unclear, right? Then with that, I think we can finish for today. Okay. You can ask any question if you have. If you want, you can take a screenshot of the board for your clarity, right? If you want, you can take a screenshot. Then I think we can finish. Okay. Thank you for joining. Any questions? If you don't have any questions, thank you for joining. Okay. Good night. Okay. Sure, you're welcome. Okay.